First, I would like to thank the organizer and the committee for giving me this opportunity to speak here. The event like this, as a basic scientist, we learn something, we learn a lot, and got motivated to work harder and work more. My job today is give a brief summary of the preclinical studies on the pain associated with TMD conditions. So this is a daunting task. I'll try to get everything. Chronic orofacial pain, or TMD pain, is a major health problem. And we don't have much solution for this, unfortunately. So that's why we do a lot of study on this. So before I start, I want to go quickly for some unique feature of trigeminal pain processing and the TMD pain. So TMD-associated pain is like deep pain and visceral pain. It's more like not like a cutaneous pain, it's more diffuse. And it feels like a cramping, itching, throbbing, dull, and often it refers to other parts. So it refers to extra territory out of injury, and also have pain hypersensitivity. Another thing we know is different from spinal cord, which the injury input goes to a few segments, maybe to spinal cord, then go up to the brain to produce pain. In the trigeminal system, particularly for temporomandibular disorder pain, the noxious information goes to multiple segments because trigeminal sensory nucleus span a long section of the brainstem. So basically, there's two main input, not only one. The caudal VC is something like a spinal dorsal horn, analog to spinal dorsal horn. So most input goes here. And also the input goes to another place, like a VI-VC transition zone. So produce a lot of other effect. So that's one thing. It's different from TMD pain. Another thing is the gene expression profile has been shown is different in the trigeminal system. This is a recent report on the transcriptome studies. We can see actually a group of homo genes highly expressed in dorsal root ganglion, but almost nothing in the trigeminal ganglion. So this is a very sharp difference between spinal cord and the trigeminal system. And we know very little about the functional significance and pathological significance of these findings. So another thing is like CGRP is apparently lower, lowly expressed in trigeminal ganglion. So there's some differences between the two systems. So we learn from the spinal cord, but we cannot completely translate everything from spinal cord to the trigeminal. That's why we need the trigeminal models also to do our research. So for the preclinical models, we have to admit, as a scientist, we want the model to be simple and reproducible. But a trade-off is that the model may not necessarily mimic all the clinical conditions, may only mimic some or part of clinical conditions. But nevertheless, those models have generated a lot of data and contributed to our understanding, enhanced understanding of this problem. So basically, many studies use this injection of an inflammatory or allergic agent into the either TMJ per se or into the muscle to produce pain and tissue injury. So there are a very long list of this agent. This is not a complete list. Like fluorous adjuvant used a lot to produce inflammation in the TMJ. So this model mimics some TMD pain, that's for sure. But one pitfall of this model is the pain produced by all those models only lasts like from a few hours or few days, at most a couple of weeks. So not like a TMD. TMD patients suffer long time, years of suffering. So this, think about this again. Now I think this is more like still is produce some kind of persistent pain, but not necessarily chronic pain. I will come back to this later. Another major model basic scientists use is the nerve injury model. Can constrict injury, transect injury, compression. Most targeted nerve is the infraorbital nerve. This is the second branch of trigeminal nerve, and it's a pure sensory nerve. So not like a spinal nerve have muscle fibers in it. This is a pure sensory nerve. So you injure the nerve, you injure the sensory pathways. Okay. This is another major models we use. Another thing is muscle injury produce pain to mimic myogenic temporal orbital TMD pain. Can produce a masseter inflammation by inject inflammatory agent. Also can ligate the masseter muscle tendon of the animal and produce a very long-lasting pain. So these models actually is different from inflammation model. The pain associated with nerve injury or this tendon injury lasts a very long time, for months. So probably more closely resemble the chronic pain conditions. But on the other hand, that's another pitfall of our research. Most research actually spend time on the initial phase of the pain, not necessarily on the chronic phase of pain. Now we feel this is more a problem because the initial acute pain, initiation of pain is not necessarily the same as chronic pain. We need to know more about what really happened in the chronic phase. Actually, this was consistent with one of my another hypotheses. I will explain more. So this is the models. Another effort is to produce a situation more closely related to clinical condition. So like manipulate the joint loading by put some splint or get some dental implant misplaced, and or simply doing repeated mouth opening. So those kind of models really trying to model real situation. But those models is more difficult to control in animals, and you need to repeated injury, and there's some confound. So it's another problem. Relatively less effort is placed on these models. There are another models want to focus on comorbidity. There's a model produced by a colleague in our department. He produced a masseter muscle inflammation plus stress, and can induce IBS syndrome as well as orofacial pain conditions. So these are comorbidity models. So these are all major animal models we have been developed and used in the past 2 decades, and which has been very useful. Another challenging aspect of studying this area in animal models is how do you assess the orofacial pain in animals. It's not like spinal cord, it's much easier because you can just test the paw response. So, but we have made quite a progress in this aspect too. So I remember in the early 80s, this is something we have for trigeminal pain assessment, put a thermal probe, implant it, and deliver temperature. It's very invasive and it's not very popular. Now we can have a model to assess thermal response in animals. Also, we can assess mechanical response by von Frey filament. Also, we have borrowed from a spinal cord. We can also do conditioned place avoidance to test the animal's preference for the place. Now we indirectly reflect their pain behavior, because animal usually rodent likes to stay in the dark area. So if you give them a dark noxious stimulus, and the dark area will be associated with pain, they will be forced out of a dark area. So they spend much more time in the night after injury. So that's another way we assess pain in rodents. Another thing is facial expression, which developed by Jeff Mogul's group. The facial expression can be used as an index for orofacial pain, and also facial grooming can also be used as orofacial pain in rodents. A lot of effort also have been dedicated to develop those operant behavioral assay, like Roger Filigrene's group in Florida. They have this model. And the nice thing about this is the animal is trained to get some reward. But in order to get a reward, they have to go through like a thermal probe or mechanical, like a mechanical barrier to get a reward. So in the normal situation, if this without pain, they are motivated to get those rewards. But after injury, they are much less effort. So that suggests the pain. So this is another kind of pain method to assess pain in rodents. Now, the biting behavior, eating behavior is another unique way to assess pain in rodents. Okay, because when you have pain, you tend to bite less forcefully. You use less force to bite. Okay, this also this is biting stuff. I have to go through quickly. So the result is you have long-lasting pain, but mechanical pain after injury, and I'll go through quickly. It's facial expression, bite force, and also the pain can be reduced by some manipulations. In this example, we use bone marrow stromal cells to reduce the pain. So I don't want to hang on for this. No. So here's some of my thought about what we have done. I think we are constantly challenged on the relevance of the animal models by clinicians. I think that's relevant. That's very reasonable because all the animal models may just mimic part of the situation, not necessarily duplicate the chronic pain conditions. So we need to work more on this. Another is the relevance of our study plan. We apparently spend too much time on the acute phase of pain, not necessarily on the chronic phase of pain. So it's more difficult to study because you have a model, you have to wait for maybe for 4 weeks, 8 weeks, not like you can do it quickly in the first week. So we need to spend more time on really chronic phase of the model to see what's the difference between acute and chronic phases. Another thing I want to mention is we still need to enhance our understanding. Dr. Calavar's presentation actually fits very well with my thought now, because I think for the whole system to be healthy, you need a balanced regulatory system, like immune system. So the both activation or inhibition system should be balanced. In the event of injury, not only there's activation of some immune cells, but your regulatory branch of the immune system is reduced. So that's really a problem, especially in the chronic phase. Probably this reduced inhibitory program may cause a lot of problem, like we just heard from Dr. Calavar. The resolution by the body per se may not be functional. So to further elaborate, I think we studied the glial activation, glial contribution to persistent pain for a long time. And we know glia is activated or reactivated, whatever. So get hyperactivity after injury. So glia gets in a reactive state. But one thing we forgot is that there's also a reduced inhibitory regulation, like this minus or inhibitory part reduced. So that may be the problem, because in order to return your body to the homeostatic situation, to normal situation, you need to do more to foster this part, to enhance this part. So not only just suppress the activated glial or immune cells, whatever. So my point is, if you want to have a health long, you don't want to only kill weeds. That will not work. You have to first give fertilizer, let the health system grow, like you enhance, return your inhibitory control to normal. So that may be another key to our understanding and management of chronic pain conditions. So have a half minute. Here's some of my thoughts. Here's my 2 cents. Okay, how are I going to go with this? I think we need to enhance interaction between clinical and basic scientists. So basic scientists will know more about the clinical situation. Hopefully we can translate this reversely to our animal models. So get more relevant animal models for this. And another thing, we need to study pain chronicity, not only acutely. A lot of persistent pain, not necessarily chronic, I think, in my opinion now, because persistent may persist for a few days, but still, that's acute pain, still protective pain. For chronic pain, that lasts for months. So I saw Bill's slide have 3 tiers. The highest tier is persistent pain. You want to change that to chronic pain. So that's why I discussed with Ron Dubner a long time ago. He said, yeah, we don't know in any model whether they have chronic pain, but no, it's persistent. Now questions asked again, so whether persistent pain is chronic pain. So that's my issue. Also, we need to study immune homeostasis, understand the whole system, and foster inhibitory immune regulation. And also, there are a lot of data out there now. Actually, we can get in touch with those data even without doing study, because there's tons of data, big data over there, omics data. But we need to analyze them, find the similarity, find some clues, and in chronic pain conditions. So that's my thought. Okay, thank my colleagues and NIH. Do I still have time?
I'm afraid not, if we were going to have discussion.
Oh, okay.
But you can bring it up in the discussion. Thank you, Kay.